Good afternoon. Thanks for listening to my talk today. I am EugeniaZarza, a researcher based at El Colegio de la Frontera Sur in Tapachula, Chiapas. Today, I will speak about our research on fungal diversity in shade coffee plantations in the Soconusco region. This is a project that involves several researchers, mainly from Ecosur. If you like drinking coffee, I hope that the beans that you brew come from a shade coffee plantation. Coffee plantations have replaced great extensions of cloud forest around the world. However, ecological research suggests that coffee shade plantations, as opposed to monoculture, hold some of the regional biodiversity and thus can become a conservation opportunity. They hold high diversity of vertebrates, invertebrates, and plants. They promote a high abundance and diversity of natural enemies that help to regulate herbivores, weeds, and diseases. They also contribute to soil fertility and soil conservation. They promote migration among fragments that facilitate a metapopulation structure. They provide habitat for epiphytes and orchids. But what about fungi? Well, we still know very little about fungi, at least at the molecular level. So in this project, we wanted to know more about this fungal diversity. For that, we collected bark samples from coffee bushes and shade trees at 3 different heights. We extracted DNA from this bark and amplified the ID's region. We obtained thousands of Illumina sequences back, and we applied the SHIME2 pipeline, including some filtering and trimming with the ITS Express algorithm and some R analysis. We also did taxonomic assignment with the database UNITE, and we detected 3,783 amplicon sequence variants. We wanted to know more about what kind of ecological roles these fungi were playing in the coffee plantations, and to know more about this, we used the FunGild database, and we found out that in the coffee plantations we can find patrotrophs, saprotrophs, and symbionts in both kind of plants and in all the samples mostly that we collected. We also wanted to see if there were some associations between fungi, taxa, and coffee plants or trees, but we did not find any statistical difference at any taxonomical level. We could appreciate some trends in the associations of Basidium micota and coffee plantations, but not really very strong. This may mean that both plants are important for the maintenance of fungi diversity. So we wanted to know more about the guild that are present here in the agroecosystem. So using the FunGild database, we investigate more in detail what were these guilds that we could find here on the bark samples. So we were particularly interested on those that are symbionts and that form mycorrhiza. We detected several types in both plants. Some of them form mycorrhiza with bryophytes and with orchids. Some others were classified as ectomycorrhiza or arbuscular mycorrhiza. This classification is a bit wide because there is not much specific knowledge for the specific species detected, but we could see that both plants hold a diversity of symbionts or potentially mycorrhiza forming fungi. Some of them seem to appear more frequently on the coffee plants, but we need to do more investigation on this. We also see this as an opportunity to plan some experiments to test if these mycorrhiza are helping for the establishment of orchids and epiphyte plants on the coffee or the shade trees. This really will help for the conservation of some of the orchids that are critically endangered. This will give further support to the idea of coffee shade plantations as part of conservation strategies. And lastly, I thank your attention, and I would like to steal some seconds to advertise a job opportunity, a postdoc position that will be available in our lab very soon. So if you are interested, please contact me. Thank you.